Hello everyone, my name is Catherine and I'm delighted to welcome you to this webinar today on the challenges and approaches in 3D ligand-based drug design. So now it's my pleasure to introduce you to the speakers for this webinar. I'm delighted to be joined by Ajay and Anne from our new BioPharmics division. Ajay is the VP of Research. He's got a PhD in computer science from Carnegie Mellon University, following degrees in biochemistry and in computer science from the University of Minnesota. Ajay has over 30 years of experience working in the field of computer-aided drug design. He has held a series of senior scientific roles within multiple biopharma companies, beginning with Aris Pharmaceutical in 1992, where he and his group pioneered new approaches to ligand and structure-based drug design. He was also faculty at UCSF for over 2 decades, with a broad research program including both CAD and bioinformatics. Ajay founded BioPharmics LLC in 1998, which became the BioPharmics division of Optibrium in August of this year. Anne is VP of Application Science for our BioPharmics division. She has a PhD in microbiology and cell biology from the University of Illinois, followed by work in biochemistry as a postdoctoral fellow at UCSF. She moved to biopharma, beginning with Metazen in 1997, leading many aspects of biological research and complex assay development across multiple projects. Anne's research focus shifted to the causal linkages between molecular structure and human pharmacology. Her research spanned both the academic environment of UCSF and the applied realm within BioPharmics LLC. Anne has a deep understanding of how to solve challenging problems in preclinical pharmaceutical discovery using sophisticated computational approaches for ligand and structure-based predictive modeling. So with that introduction, I'm pleased to be able to hand over to Ajay to kick things off.
All right. Thank you, everybody, for joining the webinar. We're very excited to have become a part of Optibrium. Instead of just having 2 colleagues between the 2 of us, we now have close to 70, and it's been great feeling to joining the Optibrium family and having now so many people to work with and interact with. So today, what we want to talk about is sort of broadly what the challenges are in 3D ligand-based drug design, but then also talk about some of the solutions. And there's also going to be a demo. And let's see. Okay. So the central challenge has to do with the fact that molecules actually don't really look like they do in 2D. So on this slide here, what we're showing are 2 estrogen receptor antagonists, which on paper, when you draw them on a whiteboard, look very, very different from one another. Turns out that if you look at how this molecule on the left is bound to estrogen receptor, that's shown in magenta over here, and adopts a very particular conformation. Those of you who are familiar with ER will this is where the steroids usually sit, and then the throat over here is where antagonists will bind, typically interacting with a carboxylic acid up over here. 4-hydroxytamoxifen is a competitive inhibitor. It binds in exactly the same place. And what you see is that it occupies exactly the same volume, but it does it in sort of a different way, where no atoms really sit on top of each other. There's maybe a couple in here, but what you're going to see throughout the presentation here is that ligand comparisons can't involve trying to literally overlap atom centers upon atom centers, because the requirement for that to lead to high similarity would mean that you have the same scaffolds, and different scaffold is a huge part of what we do. So that's one aspect. Second aspect is that the molecules that we make in the future, right, as we're developing drugs or optimizing a lead, they actually come from a different population than the things that we know about right now. So this isn't an experiment of nature where we are drawing things from an agnostic population from the universe and saying, okay, we have a population now, and the population that we're going to see when we try to predict on things is from the same population. It's absolutely not true. And Ann and I have written a number of papers about this, showing that both on short and long timescales, that the populations of molecules, even targeted towards a single binding site, look very different from one another in very meaningful ways. So simple methods to try to calculate similarity or predict how things are related to one another, they actually are going to fail to capture what underlies the biological function, which is in our case typically a protein binding pocket presenting itself and a ligand finding a way to bind into it. Last thing that is a challenge is that we often have very, very limited data, maybe a few hits or a single lead compound, or we have a few dozen compounds to start lead optimization. If you have many, many thousands of compounds, you might be able to take advantage of simplistic representations of molecules and induce a representation that allows you to learn or compare. But if you don't, it's a bad idea to pretend that you can learn from just a small amount of data, unless you're doing things that very closely mimic the physical basis of protein ligand binding. Okay, we're going to cover a few different areas broadly. One is the theory behind our method of molecular similarity calculation called eSim. And we're going to go through the physical basis of it and how that matches what a protein sees. We're going to talk about virtual screening, both using a single ligand as a query or target and using multiple ligands overlaid with one another as a target for virtual screening. Ann's going to show how that's done in StarDrop with a little live demo. One of the unique things about what we do with eSim and what you can access in StarDrop, but also directly from the BioPharmics platform, is we can do multiple ligand alignment and do it fairly quickly. But it's also actually quite accurate in terms of the poses that you predict, and we'll show you some data on that is actually not published quite yet. This approach to multiple ligand alignment is what underlies what we call Quanza, which is our method for quantitative prediction of binding affinities, and we'll get to that if we have time towards the end. Okay, so in terms of 3D molecular similarity, it's a pretty old field. I've been doing things in this area since the early 1990s. Things like rocks are widely used and have been around since the early 2000s, or actually I think even the late 90s. So there are sort of two fairly dominant ways of thinking about how you characterize surface shape. One is by looking at atom-centered Gaussians and computing field overlaps between, say, a representation for benzamidine and aminomethylcyclohexane. So these are two different inhibitors that are competitive serine protease inhibitors, trypsin-like ones. When they bind trypsin, they look like the center panel over here. And so you can imagine that if you put spherical Gaussians at the center of each of these atoms and you compute an overlap between the two, that's going to do a reasonable job of overlaying these. If you optimize that, what you see in the field overlap is that what happens is that if you're trying to calculate an optimized shape overlap using atom-centered Gaussians, that you actually end up literally overlapping atoms. And so if you take a slice through the field overlap and you look at the sort of density of the function, you see a recapitulation of the underlying atomic substructure. Another way to do it, and the way we do it, is by looking at molecular surfaces instead of looking at atom centers. If you do that, you end up with an optimal alignment over here based on pure shape, which is trying to make these two molecules fit into an identical pocket, okay? And what you see in the overlap section that we have here is that we're responding to the outside surfaces. So this is sort of what the protein sees. When trypsin sees these two molecules, it's not seeing atomic centers. It doesn't really care where particular carbons are. What it cares about is where the van der Waals surface is of the molecule. So what we see and what we're going to argue, and I hope convince you of, is that this approach fundamentally of looking at surfaces is a better match to what you see physically when proteins and ligands interact with one another. So how does that work? The way it works is we're going to use 2-pyrrolidone as an example here, and the cartoon in the lower left explains it fairly simply. We're going to put observer points around the molecule, and this is going to be our query, the magenta molecule here. And at each one of these observer points, we're going to measure several different things. The way we capture the shape, the surface shape of molecules, is by measuring the minimum distance from each one of these points to the surface of the molecule. So in this case, that's 4 angstroms. We're also going to capture the Coulombic aspects, the partial charges of the molecule, by looking at what it costs to bring a positive charge in from infinity to each one of these points. So if we do that down here, we have a slightly positive charge, so the Coulombic energy is plus 1.2. So we have a Coulombic field representation as well as a pure shape in these first 2 numbers. The next thing that we do ends up being very important, because you can't really explain hydrogen bonding purely based on partial charge effects. And so we're going to measure both the distance to the nearest hydrogen bond donor, but also the directionality of the extent to which this proton, for example, points to this observer point is going to result in a theta. Here it's 140 degrees or so. And we do the same thing for acceptors. So we have this representation now of this particular pose of this particular ligand, okay? And then we can optimize the presentation of a new molecule into this exact set of observer points and try to maximize the similarity of what we're reading out in terms of the features at each one of these observer points. So these are actually Gaussian functions that reward similarity as opposed to a difference function that penalizes differences. And so what you see here are a few different examples now of optimal alignments of different ligands. And you can see that, cyclopentanone is extremely similar. It's just missing the donor over here. Pyrrole is also very similar, where it's missing the acceptor. And this is the rank order goes like this, with benzene being the worst because it has no no sort of partial charge variation, because it doesn't have any hydrogen bond donors or acceptors. So that's the sort of theory of what we're actually calculating. The way we actually do the optimization is it could be a 3-hour talk. It's very complicated to try to do this and get answers very quickly. Conformational search is done before we apply eSim using the 4th-gen methodology. We use a geometric hashing approach to rapidly align individual conformers of our subject molecule onto our target. Here we're showing an example that's a PARP1 inhibitor, again in magenta. We then do some fast approximate scoring that mimics the full eSim function. We identify good candidates from that. And then depending on how fast we want the method to run, we'll do various levels of fine-grained optimization of molecular pose to really optimize the exact match of the cyan molecule onto the target. What we're seeing here is a depiction of the similarity function. The little gray spheres are where the surfaces of the 2 molecules match, and you can see that across the warhead in the upper left here, they have essentially a perfect match. There are a couple of areas where the molecules don't have a perfect surface match to one another. They're actually quite a bit different. You can see that we have directional hydrogen bonding. These 2 donors line up perfectly, as do the acceptors. And so that's depicted with the blue and red sticks. And then we have the Coulombic component of the similarity function shown in these spheres that are outside of the van der Waals surface, where the red spheres are saying that we have negative charges that agree. Bright blue spheres suggest that we have a positive charge that agrees. And then the mid-range between sort of purpley ones are where we have neutral charges that agree. So that's sort of what the function looks like. This function agrees very closely with what you see when ligands are bound to proteins. And what I'm showing you here is a CDK2 example where we've got, it's on the order of 32 CDK2 inhibitors superimposed as they bind CDK2. And what you'll see the characteristic sort of kinase hinge binding donor, acceptor, donor. And one of the things you'll notice when you align lots of ligands on top of each other from crystallography is you don't get exact atomic overlap. Almost none of these atoms line up exactly with another buddy. So with these different inhibitors, even if they have the same core scaffold, they tend to shift a little bit, again, because proteins don't care where your atom centers are. Proteins care about your surfaces. So the game here, typically in docking, you have a new ligand, you want to predict a pose. We use eSim to help us predict poses because eSim agrees with what proteins see. So if we take this ligand 2XNB, which was synthesized long after the pile that we see over here, and we try to predict the bound pose using docking. If we use pure energetics, we actually get the pose ranking wrong. This family up here is about a half a kcal or so higher ranking, better ranking than the correct family down here. And anybody who plays with kinases or knows them would see this bottom one immediately and say, that's the right answer. But we can use eSim to adjust our ranking of our pose family because it is caring about ligands the same way that a protein cares about them. So using an idea from statistical physics, we can calculate the similarities of each of the poses within a pose family to each of the molecules that we already know about in terms of their bound configuration, and then adjust the scores. So here we have the known pose of an earlier ligand from the structure 1FVV. And what we see is that there's a suspiciously high similarity between this pose of the ligand of 2XNB to this pose that's from an actual crystal structure of 1FVV. We have the characteristic hinge binding. We have good surface agreement in terms of shape across much of the molecule. That allows us to quantitatively score the ligands by taking into account what eSim is telling us in terms of the similarity of these poses to what's been known before. So rather than having to enforce some kind of template-based restraint, we can actually just use statistical physics to adjust scores and then get the correct pose ranking. All right, so that's the theme here of the whole presentation, is that what eSim is calculating, the underlying basis of the computation, agrees with what proteins see and think about what ligands they get to talk to. So if we use virtual screening, we've got a target ligand over here on the left, the same example that I showed you earlier, and we want to take a large number of things, hopefully that contain some actives, and we want to rapidly optimize the poses of a database of molecules onto our query. eSim is very, very fast. Using the laptop that I'm presenting on right now, it's an XPS 15, so nothing fancy, nothing exotic. We're looking at about 1,500 molecules per second, or 130 million molecules per day, just on a laptop. And so it's essentially fast enough to screen anything that you're going to want to screen, no matter how big your database is, even on just a laptop. So that's important. We have different levels of accuracy. Using the medium setting for screening, we're looking at 24 million molecules a day. If you want to use the most accurate level for screening, you're looking at 7 million molecules per day. So again, very fast, no GPU, no exotic hardware, nothing fancy, just a regular laptop. Another unique thing about the eSim methodology is you can use a set of mutually aligned ligands. So here we've got 5 ligands of LCK. It's a kind of a weird kinase. And these are the crystallographic poses of the 5 ligands. And eSim is going to look at now, take a subject molecule and try to align it to these 5 things. But the subject molecule can say, you know what, I look like this part of the yellow guy over here. I look like this part of the cyan guy, including, say, the spheno that protrudes sort of south. And then I look like the right-hand side of the magenta guy. So it's not trying to individually maximize similarity to any particular molecule. It's trying to use pieces and parts of them in order to make itself look like what the binding pocket will accept. So there's a lot of information in a multiple ligand alignment, and that's what eSim's using. If we use the single ligand up here on top, we actually do fairly poorly in terms of enriching for actives over decoys. We get an ROC area of 0.6. If we use the alignment of these 5 crystallographic ligands, including this sort of oddball, we increase our ROC area to 0.84, which is quite respectable. It's only about 3-fold slower than using a single ligand as the target, so it's not linearly bad with having N ligands mutually aligned. And so now what we're going to do is hand over to Anne, where she's going to show you how this runs inside Stardrop with the eSim 3D implementation. The thing that's really cool that she's going to show you is you don't even need a protein structure. So what I showed you with LCK there were crystallographic ligands. You don't need that. You can actually do a multiple ligand alignment using eSim and use that for virtual screening. So Anne, why don't you go ahead and share your screen and walk through the demo.
Okay, so what you're seeing here, this is the eSim 3D module within Stardrop. We're going to demonstrate today two of the most important functions of eSim, and that is, as Ajay said, multiple ligand alignment and virtual screening. So if we want to align, let's say 5 ligands, we can just go and grab them. In this case today, we're going to load them just from SMILE strings, and you'll see your molecules appear over here in the window. You just then, let's see, okay, you just select the molecules. In this case, we're going to align all 5 molecules. These are 5 PARP1 ligands. And you just come down here and you choose Generate Binding Hypothesis. The reason that we use the word hypothesis is when these molecules are mutually aligned, it is a hypothesis of their relative pose in the same binding pocket. We're just going to leave this on the default options and choose Finish. So what's happening here is there's a 2D to 3D conversion, and then a conformer generation, and also the alignment. So this is going to take less than 30 seconds on my machine, but while that's running, let's actually look. All of these ligands that we chose here today have been co-crystallized with the PARP1 protein, and what you're seeing here is the relative binding poses of the ligands that are being aligned now. What you see here on the left is what is considered the warhead of these ligands. And these elements over here make important hydrogen bonds. You can see this amine here on all 5 ligands interacts with this carbonyl on the protein. And likewise, this carbonyl on the ligand interacts both with this amine and this hydroxyl. There's more diversity on the right-hand side of these ligands. And in fact, one of them, you can see the one that ends in the terminal trifluoromethyl, actually explores a different part of the pocket than the other ligands. So our hypotheses are done. We have 10 of them. And what you're seeing here is that mutual alignment, especially and most importantly of the warheads, looks exactly like the crystallographic overlay. One thing that's important when you're doing purely ligand-based alignments with no protein is that sometimes you will get, for example, you see that this amine here has been aligned with this amine, whereas in the crystallographic pose, this is actually not aligned. That is not a problem with this overlay, and as I will show you, that this hypothesis is perfectly good at accurately retrieving PARP1 actives in a virtual screen. So let's say that so now what we can do is we can actually look. There's a nice display feature in Stardrop where you can look at the similarities of these molecules. So we're going to show the surface. In that case, that's the steric overlay of the 5 molecules. But what is interesting here is if we look at the hydrogen bond donors, you can see the similarity on the surface of these and also the hydrogen bond acceptors you can see. And so this is an important feature so that you can know what the similarity of these molecules is just visually by looking at the display. So what can we do now with our hypothesis? Well, we can use it as a target of a virtual screen. So let's see. Okay, so now we would just come down here and we would say that we want to run a virtual screen. We can just go to our hypothesis window and say, okay, I'm going to use my first hypothesis, and there it is, and you can see it in this 3D window. And next, you simply choose the database. In this case, we have a pre-prepared for this example 1/10 of the enamine stock collection The full enamine collection is 3 million molecules. This is 300,000. And so you would just choose this. The other, we would run it on good accuracy, highest speed. There's also an option to improve the accuracy of it. You can, this is the default relative volume. You're basically saying that we want hits that are approximately the same size as our target ligand. And in the examples we've run here, we've chosen to return 0.01% of the hits from the screen. And then you would just click finish. That screen has been pre-run here. It took approximately 8 minutes on my machine, which means that the full enamine would be 80 minutes here. So let's look. Now, if we look at our first hit up here, we can look at the display again. And in this case, we're going to view it side by side. And you can see that this first molecule here has that very nice similarity to the hydrogen bond donors and acceptors on the warhead, and even more similarity even in the right-hand side of the molecule. We also ran this with a single crystallographic target, not instead of the multiple ligand alignment. This happens to be the single crystallographic DUD-E target. And you can see that it too found positives with the similarity to the hydrogen bond donors and acceptors in the warhead. Using the single crystallographic ligand, it took about 3 minutes to run, which means that it would take only 30 minutes to run the entire 3 million enamine set with this ligand. I want to mention that one of the good things about being able to use multiple ligands as the target is because you have more binding information, that you actually get more efficient return of your active molecules. And so you can see that you have highly similar active molecules here using the 5 molecule target, whereas with the 1 molecule target, it worked very well, but the 5 molecules are more efficient. And so in closing, I just want to mention that these pre-searched databases, the eMolecules collection, the enamine, and the MolPort are available for StarDrop eSim 3D users, and they're pre-searched, and you can simply download those. So now I'm going to hand it back to Ajay.
All right. So as Ann mentioned, you can download all of these screening collections. They've been pre-searched and run very, very quickly on non-exotic hardware. So you can just go ahead and start finding new hits quickly. So now what I want to focus on for the middle part of the talk here is actual quantitative benchmarking of the eSim methodology. And there have been a couple papers on this, but one in particular, the first one that introduced the methodology, made use of the DUD-E set from Brian Choquette and John Irwin's lab, which was constructed as a docking benchmark involving 102 pretty diverse protein targets that included kinases, hormone receptors, proteases, a bunch of different things, where in total there were about 23,000 known actives for across the 102 targets. And then they did a process of identifying 50 property match decoys for each of the known actives from the zinc database in order to come up with a decoy collection. So the game here is to try to run some kind of screening methodology to quantitatively separate known actives from these decoy sets that were constructed to mimic the physicochemical properties of the actives. You can obviously do this, use this for docking benchmarking. That's what it was designed for. You can also use it for ligand benchmarking, and it's become a standard essentially in the field. One peculiarity here is that you can't really use it to benchmark 2D similarity methods, because the design of the decoys was intentionally done to eliminate things that were 2D similar to the actives. And so basically all 2D methods work well on the DUD-E set if you're looking at benchmarking virtual screening. So 3D methods, I think it makes a lot of sense. So for estrogen receptor, this is the example I showed you earlier. The target is the ligand of 1SJ0, and at the different levels of screening, from the fastest to the medium fast to the most thorough here in cyan, you can see this is a standard receiver operating characteristic curve, and we characterize the area under these curves. In the paper, the bulk of the results are reported at the middle level, this green curve over here. All of these are really, really good, where you're looking at extreme enrichment of actives versus decoys in the early part of the curve. Okay, across the 102 targets, we were able to compare eSim on 3 different speed settings versus ROX color, which is how most people would run ROX, with identifying donors and acceptors and so forth. And then ROX shape only, that was carried out by another group. And then there were several other approaches that go pretty fast, 5,000, 6,000 molecules per second on a single core, but they essentially perform close to random. So with ROC areas, if you're at 0.5, that's a random result. These ones actually had close to a majority of cases where they were returning random answers. So even though they're fast, we wouldn't argue that you should. We would argue probably that you shouldn't use these methods. You probably shouldn't even publish these methods if they're only producing random results. We have a very old methodology that's faster than 200,000 molecules per second on a single core. Even that performs better than these methods down here. This is using a vector-based 3D representation of ligands, and you can look at those papers if you'd like to. Importantly, eSim at all of its speed settings performs substantially better than ROX. The p-values are extreme here because there's a large number of targets, so p is less than 10 to the minus 9th in terms of the advantage that eSim on its fastest setting has over ROX. And this is across all 102 targets. We're looking at 275 molecules per second on a single computing core, so not using any multiple computing cores at all. All right, so that's using the 102 ligands that were curated by John Irwin and Brian Choquette for the cognate ligands of the proteins that they use for docking experiments. As we mentioned earlier, you can use multiple ligands to run a virtual screen using eSim. So Anne went through for all 102 targets of the DUD-E set and constructed the DUD-E plus set by identifying the set of molecules, the set of crystal structures that existed for each particular target, doing a multiple alignment of the proteins, and then identifying the 5 that captured the protein conformational variation the best. This is all done automatically using a method we call pSim, that's local protein binding pocket similarity. So now for 92 of the 102 targets, there were at least 5 cognate ligands. And what we did was run the same experiment as before, but use the 5 ligand overlay using the crystallographic coordinates for each of them. And what we see is that across the board at all the different speed settings, using a multiple ligand alignment from crystallography performed significantly better than a single ligand, okay? And in fact, if you cheated and you ran each of the 5 ligands separately and let God tell you which one was going to be best, that's what you get across the middle here. So if you cheat and ask an oracle which is the best ligand to use, you don't do better than using the joint overlay. So our recommendation when we run eSim, if you have multiple ligands, we really argue that you should use at least a few in their mutual overlay in order to improve your screening enrichment. Okay, so now, so again, I've been arguing this whole time that eSim is making its predictions for the right reasons, that it's related to physical reality. So in this joint paper that we did with Steve Johnson at Bristol Myers Squibb, we wanted to quantify that. So the way we did that was by looking at the full set of ligands that are bound to each of the 92 targets in the DUD-E plus benchmark, which allowed us to identify nearly 400,000 pairs of ligands where we know the relative crystallographic alignments of them, as we do with this example from estrogen receptor. we curated this so that each one of these things is at least nominally tractable. Each pair of ligands has to overlap at least a little bit. We're going to use one as an alignment target, and we're going to take a randomized conformer of the other one as the subject. We're going to do the eSim alignment and then quantitatively ask how well the RMSD of the predicted poses matches for each of these roughly 400,000 test cases. One of the things that's interesting about this particular benchmark that we created is that it's divided into easy versus hard cases. So there's easy cases where you might have two ligands where they have very similar scaffolds and all the atoms land on top of each other. What we have here is a hard case where we have two competitive ligands of, again, the estrogen receptor. This is the agonist binding pocket where usually you see steroids, where in terms of atomic overlap, atom centers actually co-occurring on top of each other. Only 9% of the atoms have a direct atom-atom overlap. It's only the oxygen on this hydroxyl over here. You can also look at the extent to which the molecular volumes overlap. This is 91%, so we have high overlap of these molecules, but with very little direct substructural atomic matching. So over now, all that set of 400,000 molecules, what we see is that roughly 80% of the time, eSim is able to give you an answer within the top 20 return that's within 2 angstroms RMSD. So that's roughly as good as cognate ligand redocking. So that's really impressive that just using pure molecular similarity, we're able to quantitatively predict poses. So for the harder cases where we have very little substructural atomic overlap, that drops down to 70%. Where you have more significant substructural similarity, that goes up to over 90%. We did a very rigorous comparison with rocks, and here we're showing you all 400,000 pairs. X-axis is eSim, Y-axis is rocks, and you see that rocks make some very, very crazy alignments, 7-8 angstroms away from optimal. eSim across the board does significantly better. Even where both algorithms are doing pretty well, you can see that the rocks RMSDs are higher than the eSim RMSDs. Look, if we look at the cumulative histograms over here, rocks is able to get roughly 78% correct at the 2 angstrom threshold for best pose of top 20. eSim again gets 80%. So this is the p-value is like 10 to the minus 100th on this one over here. This is true across essentially all of the targets. eSim does consistently better. So we think that eSim is really predicting the right things for the right reasons. So over the past several months, Ann and I have been working on benchmarking the accuracy of multiple ligand alignment. And as Ann showed you, you can take SMILE strings and go from there to a multiple ligand alignment. She showed you an example from PARP with a convincing comparison to the crystallographic overlay. I'm going to show you some statistics now. The way we overlay multiple ligands, we obviously we're going to want to maximize the pairwise molecular similarity. We also want to minimize the overall volume in arbitrating between different solutions. We prefer a more tightly packed one, and we want to simultaneously make sure that ligand strain is minimized so that you don't see your ligands being bent excessively. We're writing the paper up now, but using all 92 E targets, we randomized all 5 ligands for each case. We used a very deep search with the force-gen conformer search, and then used our normal geometric level of accuracy for doing multiple ligand alignments. And then we assessed how well these matched crystallographic ground truth in terms of both absolute RMSD and relative RMSD, and that'll become clear what that means in a second. Then we actually asked, how well do the multiple ligand alignments retrieve actives and separate them from inactives compared with using crystallographic targets? Okay, so the PARP example that Ann showed you is the crystallographic answer over here on the left, okay, and on the below here is the actual comparison between in cyan the multiple ligand alignment, and then in green, the crystallographic alignment. So you can see that for all 4 of the ones on the left here, you have a nearly perfect alignment except for the disubstituted phenyl. And over here, as Ann mentioned, the trifluoromethyl compound here, we get the wrong answer. There's really no way that you would guess, if you're doing a multiple similarity optimization, that these two amines don't overlap one another when there's a low energy conformation that allows you to overlap them. So the absolute RMSD is 1 angstrom deviation. The relative RMSD, this is measuring the extent to which the correct atoms correspond with one another between each pair in the multiple ligand alignment. So that's even better. So we're getting really good correspondence between the parts. And this is what's really important in terms of screening efficiency. If you at least have the correct correspondence of atoms, you're going to do pretty well generally in terms of ROC area. So I'm showing you across the top the original DUD-E ligand 0.93 ROC area. Using the multiple ligand alignment does a little bit better, 0.95. Using the top scoring alignments, this is purely agnostic, random starting conformers. We do an alignment and we run a screen. We do about the same as all of the other cases here. And for the one that actually is closest to crystallographic, it's again, essentially the identical result. AKT1, slightly harder case over here, where the absolute crystallographic comparison to crystallography is less good. You can see some deviations here between the cyan and the green molecules. The relative alignment is extremely accurate in terms of the actual correspondence of how the kinase warheads sit. This one donor is a little bit off where it should be. Other than that, that's really nice and dead on. The right-hand side here has a slightly different absolute conformation, but it's still good enough to give you really good results with respect to virtual screening. So again, using multiple ligand alignments instead of crystallographic ones is a very effective means of doing virtual screens. So here we have the beta-2 adrenergic receptor, same story as we just saw with AKT1. A little bit loose on the absolute fidelity to the bound poses of these ligands, but the relative correspondence is extremely good, where the critical sort of the mean with the beta hydroxyl over here is aligned very nicely in a relative sense. And this again produces a very accurate alignment for the purposes of virtual screening. And in terms of SAR, if you're doing lead optimization and you look at this kind of multiple ligand alignment, it's going to more or less tell you the right things about how these different alternative left-hand sides are aligned with respect to one another. And you can use that then to look for new heterocycles in here that mimic aspects of different parts of these. So again, the important thing here is that you're getting the sub-problem solutions correct, and that's enough not only for screening, but also to drive design choices if you're looking for a bioisosteric replacement of the left-hand side over here. Quantitatively, in roughly half of the cases, we got our best absolute RMSD among the top 20 solutions returned was less than or equal to 2 angstroms. 2/3 of them had relative RMSD less than 2 angstroms in the top 20. Now, when you look at the actual quantification of screening results, what we see is that the bona fide crystallographic alignment on average does the best across all 92 cases. You'd hope that that was true. The multiple ligand alignments do nearly as well, either using the top scoring one or using the one that nominally matched the crystallography the best. So using fully agnostic top scoring multiple ligand alignment reliably gives you a good result. And it's statistically significantly better than using a single crystallographic ligand. So here, purely ligand-based virtual screening is supported better by using a multiple ligand alignment if you have a few lead compounds than what you're able to do using a single crystal structure using only ligand similarity. Okay, so just to conclude on where we've gotten to so far, what eSim does is different to other methods that are alternatives in the space of ligand similarity calculation because it's surface-based. And it's completely insensitive to where atomic centers are and whether they correspond. It's really looking at what proteins care about. We're looking at the congruence of Coulombic fields, surface shape, and directionality of hydrogen bonds, including and also the distance, obviously. there are multiple different search modes that give us a trade-off between speed and accuracy. And there's no knob twisting. You just pick how fast you want it to run and you run it. You don't have to sit there and tell it the right answer and say, oh, you have to have these amines aligned. You have to have this donor receptor donor aligned. It's going to do that for you, and it's going to do a really good job. A lot of times when it's going to disagree with what intuition might tell you, it might be right. And so we argue for people to consider and open their minds to looking at the solutions that they get from eSim. We've quantified now very rigorously the comparison of eSim for virtual screening, and obviously ROX is a competent, good method. A lot of people use it. It's fast, but eSim's actually faster, and it's actually quantitatively better, both in terms of virtual screening and in terms of post prediction. And so we think the time is now for people to change and move to what we would say is a more modern method that takes into account better what proteins are looking at when they're looking at ligands. Multiple ligand alignment is a fairly unique aspect of what eSim is able to do. And from what we can tell now in the paper we're writing, it is very frequently correct, especially with respect to the relative alignments of ligands, which is enough to drive virtual screening. So I'm going to spend the last couple of minutes here talking about the Quanza approach, which depends on the multiple ligand alignment methodology of eSim, where now if you don't have any information from crystallography, but you want to do affinity prediction, you can actually do that. So the QSAR problem fundamentally can be formulated in a very abstract way, with smile strings on the left corresponding to your molecules, and then activity values on the right. Now, if you have many, many, many thousands of molecules, you might be able to use a representation like smile strings and induce a model that's going to give you some utility in terms of predicting the activity of new molecules. However, if you have what we typically have, which is a few dozen, maybe, or a few hundred molecules early in a project, you're not going to be able to use a 2D or 1D representation of ligands in order to predict, to learn a model. So what Quanza does is construct a pocket field which looks at the quantitative behavior of the molecular surfaces of molecules, and that's why it's called quantitative surface field analysis. There are a few things that are true about how molecules bind proteins that we think it's important to embed into a machine learning method, especially when you don't have much data. One is that even if you make very small changes on a molecule here, just a couple of atoms are different, the core scaffold is going to move a little bit. It's not going to stay right on top of where it used to be. So a molecule, as you modify it and mutate it, it's going to move around a little bit in the binding pocket. If you change it a little bit more, and here we have two different steroids here bound to testosterone binding globulin, it actually, the green guy here, this is crystallographic, actually flips top to bottom and left to right with a very small change in molecular structure. This is the kind of thing that people have never gotten right in terms of even 3D QSAR. 2D is even worse. But you'll see these things happen. It happens in kinases also with left to right flips. And so understanding that your pocket itself should be defining how your ligands bind, it's extremely important. You might see non-additive behavior as well, just based on size exclusion. A lot of 3D methods can't even handle this. So this is what's going on when ligands bind proteins. We shouldn't pretend like it's some magical relationship between a string of characters and a number. And without going into any detail on here, there are a couple of papers that we published that we'd like people to go and look at. But now, given these smile strings here for benzodiazepine ligands, we can induce a multiple ligand alignment and then come up with theoretical possibilities for the alignments of all of the different molecules in our training set. We also come up with variations of each of these poses. So we have a manifold of poses for every single molecule in a training set. We're able to induce a quantitative function that lives in 3D space around a virtual binding pocket. And then to predict on new molecules, we simply fit them in the same way that we essentially do molecular docking. We can score then a new molecule here with a very, very different scaffold than what we saw in our training set, and quantitatively predict fairly accurately in many cases what the pKi is. So in this case, we're predicting 8.0. The actual experiment value is 8.3. And so that, and what we're depicting here are the interactions between this molecule here and the pocket field that you see on the left. We published a couple of papers on this. This is going to be the last slide here. I'm not going to go over any detail except to let you know that we've done this now many times with pharmaceutical collaborators. This is with Bristol-Myers Squibb data on LFA1, where we'll train on molecules that were synthesized early in a project and then quantitatively predict now on molecules that were synthesized later in the project. And we've compared the results between Quanza and FEP+. And what we see, importantly, is that generally equal to or better than FEP+ in terms of the predictive quality. And then the second thing is that the predictions tend to be synergistic. So in the cases where FEP+ is giving you good results, which is in many people's hands, it's about 2/3 of the cases, and then 1/3 there's issues with, you're looking at too much charge difference or whatever. In the cases where FEP+ works, Quanza predicts sort of orthogonally. If you average the predictions together, you do better. Okay. I think I've already talked enough about the actual conclusions here on eSim, and so now I'm going to shift over to questions, but also just paint a picture here of, we don't just do eSim and Quanza. We do conformer search. We do real space fitting of ligands into X-ray density. We do docking, and all of these things are part of an integrated platform. Okay, so we've got a couple of questions, and let me take a look here. So we have one question saying, is it possible to screen the entire enamine library of 6 billion molecules? The answer is yes and no. So it's feasible, certainly, to do the preparation of the full real space enamine library. We haven't done that for two reasons. One is we prepared the full stock collection, which is now actually close to 5 million compounds. And in the experiments that we've done, the right thing to do is to screen the stock collection, look at those hits, assay those, and then do near analog expansion in real space using 2D types of tools that enamine and others provide. That ends up being a much more effective way of doing it than trying to screen directly. The other methods that are out there to screen the enamine real database use really, really gross approximations of their similarity calculations in order to nominally say, well, I screened all X billion compounds, but the results you're going to get are not as good as applying a better method to, say, the enamine stock collection and then doing sort of your virtual expansion afterwards. Okay, we've got another question over here, which is, to what extent does conformational search explain the performance differences between eSim and ROX? Generally speaking, for small molecules, not macrocycles as we're showing over here, but for small molecules, the OpenEye Omega tool does a pretty good job of conformational search. And so the conformational search differences that we see are not driving the performance differences. What's driving the performance differences is that eSim is actually calculating the right thing. So the objective function that we use is looking at what proteins care about. Proteins don't care about atomic center overlap. And so we're not only calculating the right objective function, or a better one at least, we're doing a much better job of optimization of poses of our compounds. So one thing that you'll see, for example, with ROX is that if you have a big query molecule and you have a small ligand that actually mimics a piece of that query molecule really well, ROX will end up aligning it right in the middle, okay, because it's looking at center of mass as a key method to try to align things. We're doing a much more sophisticated kind of alignment in our methodology. Let's see, so we have a question, I think, for Anne in terms of Quanza, I emphasized that, for Quanza ligand alignment, is it possible to make use of protein-bound ligand poses, and how does the use of known poses affect affinity prediction quality? So Anne, can you address that, please?
Absolutely. So even though Quanza can run in a purely ligand-based mode, that you can use crystallographic poses, either a single molecule or the multiple crystallographic molecules like the ones that I showed you for PARP, to seed the initial alignments. And absolutely, in those cases, we see that it does improve the accuracy of the predictions of the test molecules. Although I do want to say that you can also use multiple ligand alignments of some of the training molecules to also seed those initial alignments. And in that case, even the purely ligand-based model is more efficient. So seeding the alignments either with crystallographic poses or with a multiple ligand alignment does improve the prediction accuracy.
Thank you. We've got a couple of questions about preparing databases for eSim. So as Anne and I both mentioned, we've pre-prepared enamine stock collection, e-molecules, and Molport for people. They can download those from the Optibrium website. If you want to prepare an in-house collection, for example, say a few million molecule collection, you use the fourth gen methodology. It's all documented in the manual that we provide, and it'll take about 3 or 4 days on a decent multi-core Unix workstation to prepare a few million molecules for virtual screening. You only have to do it once. Most of our larger pharma collaborators prepare their databases one time and then incrementally prepare new molecules on a monthly or quarterly basis so that they have their internal databases always up to date. And then you can concatenate the files together. So it doesn't take very much time. It's conceivable to do billions. Then you need to use a cluster of multiple computers, and we are big proponents of cloud computing. And the way we license the BioPharmics platform, we typically include unlimited cloud computing access to the methods, and it's very easy to fire off big jobs on the cloud to prepare very large databases. I think we have time to answer one more question here. We have a question on how eSim accounts for ligand strain in virtual screening and in multiple ligand alignment. When we prepare ligands for running through eSim, we use the fourth gen methodology in the upper left over here, which produces ensembles of conformers for each of the input molecules. As part of that, we identify an estimate of what the global minimum is for each ligand. And we have done experiments to say that, roughly speaking, for small molecules and small macrocycles, about 95% of the time, that's a pretty good estimate of what the global minimum is. Then when we run eSim, what we're able to do is look at the conformer as we are optimizing it onto the target that we're trying to make it look like. And we can trade off making the molecule look like another one by bending it against the force field. And we trade off kcals per mole of strain for actual similarity. And that's done both in terms of multiple ligand alignment and in terms of virtual screening. And I think that's all the time we have now for questions. And I think some of the rest of them can be answered offline. Thank you.